I'm Siqi Wang, and I'm a PhD student from the Chinese Academy of Sciences, and I'm currently a visiting scholar at UCLA in Dr. Grace Shaw's lab. And I'm very appreciative of this opportunity to be here. So today I'm gonna give you guys a brief story of the sex-dimorphic aging at the transcriptome level, which has been raised by Dr. Manolis in his presentation. Okay. So to start with, we know in recent years, more and more studies found many human complex diseases exhibit the sex differential morbidity, mortality, and prognosis. And for example, males shows a higher incidence of common colon cancer, and females show higher incidence and death rate in dementias and respiratory functions. And also in stroke or some cognitive disorders, there are also some inconsistency between females and males. And we can see many of them are age-related diseases, and we know aging is a very complex biological process that accumulates the abnormal events during human aging. So focusing on the sex differences during human aging could help us to understand the sex dimorphism in age-related diseases. Refer to the mechanisms, they are mostly thought to be related to sex hormones, mitochondria, and sex chromosomes. But at the molecular level, the known mechanisms of the sex differences are mostly resolved around the genetic variants, epigenetics, the gene expression regulation, as well as the environmental exposures. However, due to the limitation of the sample sizes, the underlying mechanisms of the sex differential aging remain unclear. We know the broadest field for the aging studies is mainly based on the epigenome, and a series of aging clock has been constructed based on the DNA methylation data and predicts the biological ages and evaluates the aging rate. And it has been reported that males shows higher aging rate than females, and tumor shows an accelerated aging patterns. In addition to DNA methylations, we know the hallmarks of aging also include the histone modification, non-coding RNA expression, and chromatin remodeling, and all of which are closely related to the gene expression changes. So what we are interested in is whether we can use the transcriptome data to identify the sex dimorphism during human aging directly. We know there are different transcriptomic resolutions, including the gene expression and splicing isoform levels. And the dysfunction of alternative splicing is closely related to many age-related diseases and cancers. And in aging or disease cells, there are a series of persuasive alterations, including the changed splicing factor levels and the spliceosome dysfunction, which could result in the abnormal RNA splicing, translation, and degradation. So therefore, our study focused on the following unsolved aspects, including the pan tissue level, the transcriptomic resolution, and the potential mechanisms. And we try to address the following three fundamental questions: the sex or age effect on the global transcriptome, the sex dimorphic splicing regulation, and the sex differences in the aging rate in gene expression level. So in our study, we initiate a comprehensive analysis over 17,000 RNA-seq samples from GTEx dataset and systematically determine how sex and age contribute to the global transcriptome in both gene expression and alternative splicing level. And we then focus on the splicing changes and found their sex-biased association between the splicing and diseases. And also we focus on the gene expression level and find some sex differential aging rate in gene expression level. So the first question we ask is, how does the sex or age affect the global transcriptome? So to address this question, we first design a new value called principal component-based signal-to-variation ratio, which measure the distance between the different groups using the adjusted equilibrium distance in higher dimensions. And we can observe that sex or age has a significant contribution to the transcriptome in most tissues. And interestingly, age showed substantially larger effect than sex. And then we can also observe more sex effect using the alternative splicing profile. And also, we quantified and compared the age effect in different sexes separately. And interestingly, we observed the larger age effect in males. So in this part, we observed the individual and combined age or sex effect on global transcriptome, and also observed some certain degrees of heterogeneity between different transcriptome resolutions of gene expression and splicing and so forth. So to determine which genes or splicing events contribute to the sex dimorphism during human aging, we identified the age-associated genes or splicing events separately in females and males using the linear regression model. And intriguingly, we found that the age-associated genes are significantly overlapped between females and males, whereas fewer overlaps of the age-associated splicing events. So next, we focus on the gene expression and alternative splicing level separately to find their functional impacts. So the next question we ask is, how is the sex-dimorphic splicing regulation during aging? So we first defined the splicing events that significantly affected by aging only one sex as the sex-based age-associated splicing events, and we call it S basis. And after the functional enrichment analysis, we found the S bases are significantly enriched in the pathways such as the amyloid, beta formation, and cytoskeleton, and disease ontologies such as the cognitive disorders, which are very important biomarkers for some neurodegenerative disease. And after analyzing on the Alzheimer's dataset, we found the male-based S bases have significant overlaps between the AD-related splicing events, and we also observed a stronger correlation between the AD-associated changes and the age-associated splicing changes in males. And interestingly, this pattern could not be found in females and gene expression level as well. So to further examine the contribution of individual S base to AD, we construct a sex-stratified classifier using a subset of S bases, and we observe some important splicing events that are associated with the AD neuropathology, like plaque and tangles, and we also found the higher model performance predicted by the male-specific S bases. So in this part, we found the sex-based association between RNA splicing and diseases, which could help us to understand the complexity of the sex dimorphism in age-related diseases. So as we have shown in the background section, we know the dysfunction of the splicing factors has been reported as the hallmarks of aging, and we surprisingly found a larger proportion of the age-associated splicing factors that are female-specific. And after constructing the AS regulatory network, and we found most of the S bases were regulated by this type of sex-specific age-associated splicing factors. So next we asked which factors could regulate the expression of these sex-specific splicing factors. So we focus, mechanistically, we know the estrogen consistently decreased during lifespan and banned the nuclear receptors to control the gene expression. And indeed, we found ESR1, which encoded estrogen receptors, are differentially expressed during aging in a sex-specific manner. And also, using the ChIP-seq data from ChIP-seq atlas, we found this type of sex-specific splicing factors are preferred to be bounded by the ESR1. And after analyzing the RNA-seq samples from under the estrogen treatment and the ESR1 mutations, we also found the significant activation after the estrogen treatment. So in the earlier results, we found a large proportion of age-associated genes that are common in both sexes. So therefore, we next ask whether they are changing in the same rate. So to address these questions, we identified the chronological genes, and we conducted time series forecasting and breakpoint analysis using the ubiquitously changed age-associated genes during aging. And interestingly, we found a persuasive sex dimorphic aging rate in most tissues. And also, we found an earlier age breakpoint and a higher aging rate in in males specifically. So this result could, to some extent, implying that males could be more vulnerable to aging. Yeah. So to further examine how the individual gene affects the aging rate, we designed a pipeline to identify the aging modulated genes. We call it AMGs. And we found over 70% of the AMGs are sex-specific. And the female-specific AMGs are associated with the mitochondrial and oxidative functions. And the male-specific AMGs are related to immune cell self-differentiation processes. And we also deepened into the mechanisms based on the sex hormones. We found that the male-specific AMGs prefer to be bounded by androgen receptor, and the female-specific AMG are preferred to be bounded by estrogen receptor. So collectively, our study raised a comprehensive picture of the transcriptomic sex dimorphism during aging at the multi-tissue level. We observed the individual and combined sex or age effect, the sex-based association between AS regulation and diseases, as well as the sex differential aging rate in gene expression level. And we dived into the mechanisms based on the sex hormones to identify the regulation of the gene expression. So here is the bioarchive link of our paper. If you are interested in our paper, please check it out online. So thanks to my supervisor, Professor Wang in the Chinese Academy of Sciences, and our collaborator, Dr. Li, for his support of GTEx data. And I also want to express my gratitude to Dr. Grace Xiao for her support during my visiting period at UCLA. So thanks for listening and welcome your questions and suggestions. Thank you.
Any questions for Siqi?
I'm trying to wrap my head a little bit. Excellent talk, by the way, but I'm trying to wrap my head a little bit around these slides that you showed at the end with the aging trajectory, let's say.
Oh, trajectory analysis.
The gene. Do you see differences, I guess, in those across different tissues?
Yeah, there are some differences between these tissues. Some tissues have higher aging rates in males, and some tissues have higher aging rates in females. And we integrate all of the tissues and found most tissues have the higher aging rate in males.
So most tissues are like male.
Males.
Okay, I see. Thank you.
Thank you.
Thank you for the great talk. What was the age range of people you were looking at for the expression data?
The age range from 20 to 70. Yeah.
So with that, did you look at how the trajectory of XX individuals will change pre and post menopause? If that changes their aging trajectory from, let's say, females 20 to 50 versus in that 50 plus?
Yeah, I grouped the genes between the young group from the 20 to 40, and the female group from 60 to 66 to 70.
Okay.
And we first identified the age associated genes and use these age associated genes to analysis this time series analysis and breakpoint analysis.
Okay.
Yeah.
Thank you.
Just related to the last question, do you also know the menopause status of the women? To see if there is menopause specific effects independent of aging?
You mean the functions related to menopause?
Yeah.
Not really. But in our enrichment analysis of this age modulated genes, the function is related to the functional decline of the menopause.